Hey, AP Biology, we're going to go ahead and look at how to use the chi-square formula in genetics. We're going to start with a very simple monohybrid cross that should have a really easy to see ratio and see if our results fit that expected ratio. So expected and observed, those are some terms you probably need to just keep in your mind as we go through this stuff. So, first thing we need to do is look at the genetics information in the practice problem up there and figure out what our expected ratio should be. Okay, so we see in peas yellow seeds are dominant over green seeds, so big A, little a, and a cross between two plants, both heterozygous. They are both heterozygous for seed color. That's pretty easy. Let's go ahead and start there. Okay? That's big A, little a, big A, little a, and should be one of those really well-known ratios, okay? This is phenotypic ratio. So all of these are of the same phenotype. And then this one here is of a different phenotype. Okay, so looks like we have a 3-to-1 ratio of, because yellow seeds are dominant, these ones are going to be your yellow seeds. Okay, I'll draw a yellow seed here. And the green seeds are going to be 1. So that is our expected ratio. We should have 3 seeds for every 1 green seed. Now, if we did that by percents, that would be 0.75% or 75%. I put it right in decimal form. And 25% will be green. So we need to keep those numbers in mind. Now, I'm going to make a little table at the bottom here. Okay, so let's look at what we observed right here and start working it into a data table down here. Let's go with our phenotypes. Okay, we're going to do our yellow, and we're going to do our green. Okay, so we have our first piece of data we need is our observed. And that's going to go in this first little block here. I probably made that too small. I'm going to make that a little bigger. So this is our observed data, and that comes right in from here. We had 4,400 yellow and 1,624 green. Okay, so the next thing we need to see, that's what we observed, but what's the expected? Okay, when I add these two numbers together right here, that's going to give me my total. So the sum of those two numbers is 6,024. So that's my total. That's my total. So I'm going to take that number and I'm going to use that to figure out my expected. Okay? Give myself a little room here. So I'm going to take remember I should have 0.75 on my yellow. I'm going to take my 0.75 and I'm going to multiply it by 6024, and that's going to give me 4518. Okay, so that's my expected 4518. Let me see if I can write that a little bit neater. I'm just gonna write that a little bit bigger, 4518. All right, so I'm going to do the same thing down here. I'm going to multiply this number, the 6,000 right here, the 6,000 right here. It needs to be multiplied by 0.25. Okay, that was kind of hard to fit in there, so this time I'm just going to write 0.25. It's going to be, looks like 1506. So look, guys, these are my expected, these are my observed. Up here in the right corner, see my laser pointer? Those are the numbers that go right here. Observed goes here, and the two expected values go here. Okay, and then we're going to take the sum of all those. Okay, so we take observed minus expected. Okay, so our observed minus expected, okay, so we're going to subtract these two. And let's go ahead and write observed minus expected. So when I subtract these on this one, I get negative 118, and on this one I'm going to get positive 118. Okay, so what did I do? Subtracted this number from this number, and then the same thing up here, this number and this number. Okay, this number and this number. So now we take the difference and we square them. Okay, so we're going to square those numbers, and because they're the same, that's going to be a nice little math shortcut. Okay, so this is going to be, from the formula, this is going to be this part right here. Okay, this part right here is what I'm putting in this table. Okay, so let's do that. Just mathematically, we're going to write observed minus expected squared, and that's going to be a pretty big number. 13,924, 13,924. All right, now we're going to plug that into observed minus expected over expected. Okay, so what's that going to mean? We're going to take this number right here, and we're going to divide it by the expected result. Okay, so what we're going to do, we're going to take this number, okay, and that's going to be the denominator. Let me write the formula. Okay, observed minus expected squared over expected. So where do the numbers come in? Well, here's your expected. Okay, here's E. This one right here is going to go here, and then this value here goes up here. Okay, so for this one, it's going to be 13,924. That's this term right here. See, we figured that out right here. And we're going to take this, and then we're going to divide it by 4518. Okay, and when we do that, okay, it's a little bit small to write that math in here, so I'm just going to temporarily draw it up here. We're going to take this number right here, which is this, 13,924, and we're going to divide it by expected. So where's that expected come from? It comes right over here. So this number is going to move over here. So that's 4,518, and that value comes out to be 3.08. Okay, I'm going to do that again. Okay, but I'm going to do it from a different spot. We're going to take this number right here, and we're going to plug it in right here. Okay, because we're going to take observed minus expected, that's this right here, and we're going to divide it by expected. Well, where's our expected? Our expected is over here. So what goes in this box right here? That's going to be 13,924, that's this number right here, divided by 1506. And when I do that, I get 9.24. So guys, we're going to take this number, We're going to add them together because remember, the sigma right here is the sum of all these things. So we're going to sum 3.08 and 9.24. And when I get that, okay, I'll write this one in a different color. I'm going to add those two together, and I'll put my answer right here. And that's going to be, my chi-square is going to be 12.32. So that is my chi-square value. Okay, we're going to take this number and we're going to look at how that fits into a critical value table. We're going to talk about, well, how many degrees of freedom do we have? But for right now, we have computed the chi-square value for this data. Let's take a look at how we interpret it next. Okay, so let's look at our results from that math we did in the table. We have a chi-square value, I'll just write this down, of 12.32. Now we have to calculate our degrees of freedom. So our degrees of freedom, there were two categories of data. So two categories of data, so n equals 2. And to calculate the degrees of freedom, you just subtract 1 and you get 1 degree of freedom. Where's that number important? You're going to take that number and match it over to here on the chart. If you start doing dihybrid crosses, you could get several different, you could get pretty high up on that degree of freedom table. So the area of this table we're worried about is right here. Okay. So the one thing you want to pay attention to is this value greater than or less than the degrees of freedom? Okay, is this value, this chi-square value, greater than or less than the degrees of freedom? Well, I'm just going to write this down. Okay, 12.32 is greater than 3.84. This number right here, these numbers from the table, are called the critical value. In most of our experiments, we're always going to use the 0.05 value rather than the 0.01 value. Okay, so look, our 12.32 is several times larger than 3.84. So what does that mean? That means that the data did not fit what you expected. Okay, what did you expect? You expected a 75, you expected 25% and 75%. Okay, that's not division. I'm just sort of writing down the two expected numbers. Your numbers were so far away from that data that the chi-square value got large. So when the chi is high, the results will fly. That means like fly away, they're no good. When the chi is high, the results will fly. They're just going to fly out the window. Those are no good. I hope you can remember that little memory tool. So high chi is bad. Okay, high chi is bad. Now, scientists don't like to say that data is good or bad. It just is. But this is just sort of a teacher trick here to help you remember it. So it says, does the data fit, if we go up here, does the data fit the predicted phenotypic ratio? And the answer to that is no. Okay, so we fail to reject the null or the HO. Okay, we fail to reject. So something went wrong in this little experiment here.